Can you hear me? Do I need to do a sound check or anything? Or are you?
I think we're good.
Cool. So we go get started. All right. Yeah. Well, thanks for having me today. Me and Steve we're both from Protochips, and today we're going to talk about the different types of applications, capabilities, sort of performance of some of the products that Protochips sells for in situ electron microscopy. So I'm going to try to cover a wide array of different application topics, just to try to hit on some stuff that may be of interest to you. But of course, if you have any questions or something I don't cover that you're curious about, we'll leave time for questions at the end. But for starters, I always like presenting something that I think most people who are familiar with electron microscopy are sort of familiar with from an ancillary level, which is the growth of transmission electron microscopy performance capabilities over time. So the first microscope was discovered and used in the early 1940s. And since then, the analytical capabilities of the microscope, the imaging performance of the microscope, all of that has gone up over time. And this is a log scale of just research interest over time, looking at Google Scholar and things like that. And what you can see is a growth over time. And I think everyone's familiar with how important the TEM has become to nanoscale science and sort of research and development in the 21st century. And I just kind of put a few different techniques and their effect on this publication interest. Most recently, spherical aberration correctors that kind of changed the game in terms of what was possible with STEM. But one thing that a lot of people aren't familiar with is that the first in situ publication was actually way back in the 1940s. So Ruska, one of his first papers, was doing in situ environmental analysis of the sample in the very early days. And so before we get into that too much, I wanted to talk about what in situ TEM means, what I'm referring to when I talk about that. So again, the TEM has an abundance of analytical capabilities that are just inherently built into it. So whether you're doing size analysis, chemical composition, morphology, structure, distribution, all of that information is accessible to you at all times in the TEM. But this is just sort of a one-dimensional characterization capability that you can access in the microscope. And within situ, you're adding an entirely new dimension to that experiment. So you're adding some sort of stimulus to your sample. So that could be pressure, it could be temperature, electrical bias, mechanical testing, all those different types of in situ stimuli, within or without the presence of environment, so a gas or liquid or something like that. And what that allows you to do is take an observational tool, which is a microscope, just to look at something and turn it into an experimental tool. You're actively engaging and actively recording and working with the dynamics of your sample and reactions. And of course, because this is all dynamics, you're also getting this third axis, which is the development of all of these changes over time. So you're taking really a one-dimensional data set, and you're forming a multi-dimensional data set that has a lot of dynamics, and you're coupling that with the imaging and analysis capabilities of the TEM. So that's kind of what I refer to when I mean in situ, is just taking the TEM and turning it into this experimental nanoscale laboratory. So going back to sort of the publication interest, this is something that I was curious about a few years ago, which is looking at how in situ as a technique has grown over time. So you take the total number of publications in terms of this research interest, you sort of divide out how many of those people are looking at in situ, gives you sort of a relative ratio of how important this is to the field of TEM. And like I said, in the very early stages, there was a lot of work that was done with Ruska. In the early days, just to see what kind of things they could do in the TEM. But what you can see is there was a pretty rapid decline in using that technique. People were more focused on developing the imaging capabilities of the microscope and less interested in the really finicky, difficult dynamics they could capture with experimentation. And you can see that in a paper kind of summarizing everything in 1979, essentially saying that in situ is just really difficult. It's tedious, it's hard to control, the imaging isn't all that great, things are drifting. The net summary of it is that it did not meet the criteria for true scientific research, which is that you could demonstrate reproducible results, right? So it was this finicky sort of avant-garde approach. But all of that started changing in the 2009 timeframe with the introduction of MEMS-based sample control. So ever since then, we've seen a rapid development and a huge adoption of these products because of their ability to do quantitative analysis and experimentation on samples in situ. And so the weaknesses and the limitations of products over the past decades, most of those are no longer a concern anymore. And customers around the world are now producing research like this. And so this is just a kind of a quick snapshot of the type of dynamics that you can capture with in situ, all of which is occurring within your electron microscope. So for today's talk, what I'll do is talk about the 3 products that Protochips offers in the field of in situ electron microscopy. So we have an atmosphere system, which is a gas cell, fusion, which allows heating electrical biasing, and Poseidon, which is a liquid cell. And I like putting this map up there just to kind of get a sense of the real diverse array of applications that are accessible with in situ tools and capabilities. We see customers around the world looking at applications ranging across the scientific discipline, material science to life science. And very often, and at sort of an increasing rate, we see multiple researchers who are studying one field of interest and are using multiple products to sort of serve that interest, whether it be solid state batteries with fusion, lithium ion batteries with Poseidon, or people who are using one product to serve multiple applications. And we're not blind to the sort of synergistic approach of using multiple products to serve a tremendous array of applications, which is why we kind of make these products work really well together. They sort of work synergistically so that your lab can access a lot of different applications. So what I'm going to do today, as I mentioned in this talk, is go through each of our products and just talk about some different applications, talk about how the product works a little bit, and just kind of run through some of that stuff. So first, I'm going to talk about our fusion product, which does heating, electrical biasing, and simultaneous electrothermal experiments. And these are just a few of the different applications that are sort of big hitters for fusion. Obviously, anything in material science, materials characterization tends to be a big interest. And in situ heating is, I kind of see it as like the bread and butter of in situ. It's usually what most people think about when they're talking about sample control and mimicking or changing the conditions of their sample. And that has a lot of utility for a lot of different applications. But before I go into the applications, I always like showing sort of an animation of just how it is that this is possible. So the first thing is samples are just deposited on the MEMS chips, which I have here. We call them E-chips, and I'll refer to them as E-chips throughout this presentation. They sit on top of a electron transparent silicon nitride membrane. Those E-chips are then loaded into the fusion holder. Electrical contact is made with the pen system here. And that holder just goes directly into the electron microscope. So there's electrical connections to the back that connect to our Clarity software, which I'll talk about as well in a bit. But then once the holder is assembled, it goes into the microscope, and then you're ready to do your image. So that's just kind of how your sample gets from outside the lab into an in situ holder. And now I'll kind of talk more about what you can do with that. So the first application I'll talk about is something that's near and dear to my heart, which is metallic glass crystallization. My background is actually in metals processing. And these particular class of materials, if you're not familiar with them, are really bizarre metal systems that have a lot of different application potential, from body armor to catalysis to corrosion to golf clubs. I think Apple was looking at iPhones being made from metallic glasses at one point. But they're also inherently unstable. This metallic glass phase is not thermodynamically stable. And this particular researcher was interested in even more complex subset of metallic glasses, which are nanowires that have a glassy composition. But these nanowires are not stable. They can have different crystallization temperatures where they lose their glassy properties. And in order to characterize how they could be used, they needed to change the temperature of the nanowires and see what happened to them, essentially. But because they're nanowires and they're very small, they have very little mass, you can't do traditional calorimetry. And so they use in situ TEM to not only conduct sort of calorimetry type experiments, but also visibly see what was happening in the sample and then collect all the crystallographic information, the diffraction pattern. But in order to do that, you have to be able to heat your sample up accurately. It can't move around a whole lot when it's heating up. And you also need all of the nanowires across the entire membrane to heat up at the same rate, at the same temperature. So you need a uniform and accurate membrane. And you can see here uniformity on the right, which is the glass and SEM all melting at the same time. No matter where you are on the membrane, that's the uniformity. And then down here, it's just showing the low thermal drift that you get with these. So you're getting 500 to 600 degrees, and it's just moving a little bit. I also point out they use double tilt functionality. You can see that up on the animation on the right. We build our double tilt mechanism so that you retain electrical contact with the each of at all times. So that no matter where you're tilting, there's no fluctuation in temperature while tilting.
So is it the double tilt, both directions?
Yeah, that's what I'm showing here. So this is the alpha tilt, and then you'll see in a minute the double tilt, the beta tilt, which is rotating around those pins.
So you have it both for the gel microscope and API?
We do.
Okay.
Yep. So this is just kind of the background, but the real interesting stuff is shown here. So essentially, by heating up these amorphous nanorods, they were able to initiate the rapid onset of crystallization. That can be seen visibly in the image there, but also can be seen in reciprocal space in the diffraction pattern. So in the glassy phase, you're getting amorphous rings. And then when it crystallizes, which happens rapidly in these systems, you see diffractive peaks. And you can heat up even further until you melt the sample. So they were able to image all of this in the microscope, and they were able to correlate the crystallization at different thicknesses of these nanowires. So when they deposited them, they had a lot of different thicknesses. This is a little bit of a larger nanowire. And I put here that as temperature goes, as the nanowire gets smaller, they become inherently less stable, but then there's a reversal. What I mean by that is shown here on the graph. So as the diameter gets smaller and smaller and smaller, the nanowires become inherently less stable, forming this crystalline phase at lower temperatures, but then that trend reverses, as is often the case in nanomaterials and nanoscience, that when you start getting very, very small feature size, other factors start governing the kinetics and the thermodynamics of the system. The interesting thing is that the reason for this occurring in this particular system could be visibly seen directly in the microscope. So this is a nanowire that has 20 nanometer thickness, but it also features a neck region that's even smaller. And what you can see is the crystallization front passing through the nanowire and visibly slowing down in that neck region. So this is a physical manifestation of the reduction of energies necessary to drive these different crystallization fronts. So propagating slower, and there's a lot of thermodynamics and kinetics behind this that I won't go into, but it's pretty cool to be able to physically see a mechanism that you can analyze and understand through other mechanisms, but you can actually physically see it in the microscope.
So how much of this that we are seeing here is because of these, if you have any gradient in the temperature between the two sides of the nanowire, and how much of it is the physical property of the nanowire itself? So how can we differentiate? Is it for the specification of your chips in terms of the thermal gradient?
Yeah, no, so we calibrate every single chip that goes out the door. So we're in our analysis, we're looking at the thermal accuracy, how is it the correct temperature, but also the thermal uniformity. So no chip has ever been used in a fusion system that's not gone through that calibration process, which is why we stand behind the thermal uniformity that's necessary to do these types of experiments. Because you're right, if that nanowire was one chip temperature on one side and one temperature on the other, that wouldn't work. But that's why it all heats up uniformly. So moving on to a different application, I'll talk sort of broadly about are the synthesis and characterization of hybrid nanoparticles. So these are nanoparticles that have multiple phases, sometimes complex structure, often very different chemistry. And they can be used, and they're being investigated for their utility in a lot of different applications, fluorescence biolabeling, photocatalysis, quite a few others. In this particular case, they were interested in synthesizing complex heterostructures of these nanoparticles by converting them in the microscope. So in order to do that, they wanted to heat these nanoparticles up and observe phase changes in different dynamics at high temperature. But because they were alloyed, they also needed EDS analysis to characterize the change of the chemistry of the nanoparticle over time. But also, which is very important in a lot of these experiments, is to have an inert heating membrane. So that's a membrane that's not interfering or changing the sensitive chemistry that you're often trying to understand. So you need an inert heating membrane. And this actually is, to your point, this is an example of an actual pyrometry measurement we do on a thermal chip. So this is a real data set from a real chip, and every single chip goes through the same calibrations before going out. But again, I'll fast forward through some of that to get to the interesting part, which is what they actually saw. And in essence, there is a very complex chemical pathway by which these nanoparticles essentially invert. So they start with one structure, and then they go through this alloying process, and then they invert into a different structure. And you can see down here at the bottom, the EDS and elemental analysis showing the formation of a gold-rich surface and a silver oxide-rich interior. And there was a lot of different chemical pathways that they sort of proposed for how this could occur. And they were able to mimic some of those in the microscope by controlling the temperature, but also by using the electron beam to initiate different reactions. So this is just one example of nanoparticle synthesis. And another one I'll talk about is a different material system, but a very similar research effort. So in this particular case, they were looking at another bimetallic system that went through a different pathway, but still involved two different alloying elements and their interplay as the temperature of these nanoparticles is increased. And I kind of cheat and show a little bit of the end result here, but this is some ES analysis showing the development of these nanoparticles over time. And one thing that I wanted to point out here is, of course, if you're studying nanoparticles, you still need atomic resolution, you need EDS analysis, you want to make sure the membrane is doing what it should. But as is often the case with some of these longer experiments, or oftentimes if you're repeating experiments, it can get really cumbersome to have data sets that are from your in situ experiment, but then have image sets over here and videos that are over here. And tying them together can often be very difficult. We recently released a new set of software that ties all of the experimental data that comes out of our systems and tags it directly into the images and videos acquired with a GTAN camera. So all of the data is just automatically logged into the metadata, and you'll see an example of what it looks like on the next slide. So doing in situ post-processing is not necessarily easy. We're not blind to that. We spent a long time just developing software so that you didn't have to have that problem in the lab anymore. So these researchers took that software and plugged just temperature, for example, onto the nanoparticles that they were studying. And so what you can see is as the temperature increases, these core shell particles start segregating, forming these two different alloying, or two different elements on each side of the nanoparticle. And as temperature is increased even further, you'll see sublimation of one of those phases. And the resulting nanoparticle is shown on the right as more homogeneous. And you can see here the atomic resolution mapping at high temperature of this nanoparticle that they were imaging. So they were able to do crystallography on it, identify the phase, and even pull out a few dumbbells at very high temperature. So this is just one example of some of the nanoparticle chemistry and reactions that you can see in the microscope. So moving on to a totally different field with memory devices. This is not my area of expertise, so I'll go through this pretty quick. But in terms of next generation memory devices, characterizing them, there's a lot of challenges to be overcome. This particular researcher was looking at conductive bridging resistive access memory, which have they're a lot more efficient in terms of the energy consumption utilized to make them work. But then the mechanism by which they're reading and writing and conducting memory applications is shown here up on the top right. So as a voltage is applied, a conductive filament will grow between the two conductive layers, and that will form an electrical connection. And they wanted to characterize that in the microscope, but they also wanted to be able to control the growth of that filament by applying the voltage and measuring the response of it. So in order to do that, you need accurate electrical characterization. I'm showing here just what an electrical chip looks like in the microscope. We have tremendously sensitive measurements that we can conduct with fusion. They also wanted to track the chemical composition. On the next slide, I'll show you an example of what the actual device looked like. But as is often the case with a lot of material science applications, but specifically in semiconductors, they need to do bit prep. And so we recently actually have developed an entirely new set of chips for electrical characterization specifically that allows any bit sample process to basically swap out the copper grid, be replaced with our electrical E-chip, and you can see the landing stage of the lamella here, where you're bringing the lamella in, attaching the sample to the E-chip, and now you've got an in situ experiment ready to go. You can do final thinning on the chip. You can do everything that you normally would do on a copper grid, but it's just on our E-chip instead.
So your metal plate, they're freestanding. There's no silicon nitride under the metal.
Correct.
Okay, so that in order to avoid charging.
Yep.
Okay.
Yeah, so there's a lot of benefits to this. The first is that it's a lot easier to prepare samples than it would be with the membrane in the way, so to speak. But you can also, you have a lot better control of final thinning. You have a lot better control of securing the sample to the electrical contacts, and that has some implications for electrical characterization, being able to do that more effectively. But what I mentioned before, which I'm pointing out here, And on the top right is a cross-section schematic of the particular device they were interested in characterizing. So you can see the multiple layers here, but what they were really interested in was that sort of middle layer of tungsten there. And so what they were able to do is attach that sample to the electrical E-chip, do final thinning. They had to create an electrical short so that they could conduct the biasing across that transistor. And what you can see here is the EDS analysis before and after the device was pushed to failure. So these devices, again, I'm not an expert, but there's multiple stages they go through for the read-write when voltage is applied. And that's shown here on the left. But when they pushed the device all the way to failure, they were also interested in how that occurred from an elemental standpoint. So what you can see with the arrows shown here is copper segregation, the development of an oxide across 2 layers, and then the migration of titanium around some of these layers as well. So there's some chemical migration that occurs, which has implications for the device failure. But really importantly, and I don't show it here, they were able to take the same measurements that they conducted in situ and compare it directly to the same type of experiment that was done ex situ. And the two showed a very, very strong correlation. And part of the reason that was possible was because of the capability of doing a lot better electrical contacts with these particular E-chips. So there's a lot of benefits to this particular product, which we launched just this year. But yeah, this is just an example of sort of a new age memory device and how you can characterize it in the microscope. So moving on, I think this is the last application I'll talk about, are 2D solar cells. So this was some research that was done at Oak Ridge National Lab. They were interested in some complex 2-dimensional materials, utilizing them for next generation solar cell applications. And in particular, what they were interested in is the edge structure of these 2D monolayers, essentially. So in these 2D materials, the edge can have a profound impact on the final device performance and properties. And so they wanted to characterize some of that, and they wanted to be able to control the temperature and other things within the microscope. As you'll see in a bit, they needed atomic resolution because this is a 2-dimensional material. There's not much other than atoms there. They needed a high thermal accuracy so they could heat it up and control. And then in particular, they needed a neon compatibility. So there's neon microscopes aren't as predominant as some of the other OEMs, but we worked closely with NION to develop a holder for their instruments. And so when I mean edge structure, this is kind of what I'm referring to. You can see here the black vacuum on the right. When they were heating these samples up by controlling the electron beam, by controlling the sample temperature, and by finding areas that had more or less carbon contamination, they were able to synthesize, engineer, characterize, and correlate the edge structure at atomic resolution at these different different conditions. And you can see what that looks like here. Now, these edge structures would have implications for the magnetic and electronic properties of the 2D materials. And they didn't, in this particular paper, they didn't talk as much about the properties that would be governed by these types of edge structures so much. But they did talk and they did show in exquisite detail how they were formed and how they could manipulate and control the environmental conditions to synthesize and engineer different edge structures in monolayers within the sample. So this is just an sort of schematic video showing how some of these pores and defects and edges are evolving over time, and also the complex interaction between various edges and various defects.
Is it at elevated temperature? Or
yeah, this is at 500 degrees C. Yeah, and another example of that is shown here. So all this was done at 500 degrees C. And they were able to see the etching of these edges terminated by nanowires at various carbon contamination levels. And there were some implications for the diffusion rate of various alloying elements. But in general, I like showing this just to sort of show the tremendous capability of imaging 2D materials in situ and the sort of device control that you have when you can control even just temperature in the microscope.
So this is on the whole of your grid, it's not on the silicon carbide?
Correct, yeah. So just in conclusion, and I try to cover a few different application topics, but I always like pointing out that this is a small subset of all of the different types of research areas that are actively ongoing around the world. So these are just some of the results that other researchers have published recently in the field of material science and using our products. But I also just wanted to point out a few key important variables for doing sort of in situ experiments with fusion. So, uniform, accurate heating membrane that doesn't interact with your sample, very important for a lot of sensitive chemistry. Using electrical E-chips that have low leakage, being able to accurately characterize your sample is very important. EDS is really important for a lot of reasons I think I've already stressed. But data synchronization can really help mitigate some challenges in data processing and after the fact, after going through some of these rich data experiments. And then also, as is always the case, you'll run into problems when you're doing in situ experimentation. So having a product that's been used a lot, but also having a technical support team and application scientist team that can actively help you when you're running these experiments. I think all of that's very important, so I just like pointing that out before we move on. So this was Fusion in a nutshell. But I'll move on now to Poseidon Select, which is our liquid cell system. And I'll talk about how this works in a moment, but these are just a small little scattering of different types of applications that we see. As you know, most things on Earth and most things that are used by humanity involve some interaction with liquid. Ourselves, we're liquid bodies walking around. So in biological processes, pharmaceuticals, life science, there's a lot of applications that involve the interaction of chemicals, samples, and things like that with hydrated environments. But then you also have nanomaterial characterization, corrosion, and quite a few other different applications that are accessible doing liquid analysis in TEM. But again, I want to talk a little bit about how the product works before I go into what it can do. In the case of closed cells, you're still going to be depositing your sample onto a silicon nitride window. But in the case of closed cells, Poseidon here and Atmosphere, I'll just show later, those two, you are going to be using two chips to encapsulate and seal your liquid layer. So in this case, the sample is deposited on the top chip. The O-ring gasket goes down into the holder. That forms a hermetic seal, but it also automatically lines all of the parts so that the two windows fall directly on top of each other. And then once the lid is put on top of the holder, that forms a hermetic seal that isolates the liquid layer from the vacuum of the microscope. With Poseidon Select, we've embedded electrical contacts that are isolated from the liquid, and that allows you to do electrochemistry experiments and liquid heating experiments if that's something that you're interested in doing. You can, of course, just encapsulate a liquid layer and do nothing but image the liquid if you want. But just like with Fusion, the holder goes into the microscope. We've worked very closely with Joel and FEI to make sure that these products meet their rigorous standards for safety, compatibility, and reliability. But once in the holder, or once in the microscope, I'm showing schematically here just where the liquid is going. So you don't have to flow liquid if you don't want to, but if you are doing liquid flow and mixing experiments, the liquid is going to come in these channels, go over the bottom chip, underneath the top chip, and out the back. Your sample can be on the bottom or the top window, so your sample is exposed to this liquid layer. So that's kind of how liquid gets into the microscope safely and reliably. And now I'll talk a little bit more about what you can do. So in the field of material science, one of the things we see a lot of, Poseidon used a lot for, are nucleation and growth experiments. So this particular application I'll talk about is the growth of dendrites, which is often in metal solidification, batteries. Dendrites are a common form of nucleation growth for materials coming from a liquid and growing a solid. But in order to study that, they needed a few things. One, in general, when you're doing sort of nucleation growth experiments, it's very often that you'll want to clean tubing, or you want to leave the product in good shape for the other person who may be using it next. And so we've built replaceable tubing into our holders so that you can just take them out. You can replace them, you can buy new ones, you can clean them, and they screw in. And I've got a display unit here that you guys can unscrew that and see what that looks like. It takes just a minute to unscrew one and put new ones in. I'll talk a little bit more about flow control in a bit, but it's also important when you're doing a lot of these experiments that you don't have a holder that takes hours and hours and hours to assemble. So we've made these holders as easy as possible to go together. They can go together in just a few minutes with some practice, and they always form a seal. So the last thing that I'll mention before moving on, I think I've already referred to this earlier, but when you're doing in situ experiments, you always have the electron beam as a variable that you can control. So in the particular case of liquid experiments, the electron beam can locally change the chemistry of your solution, and it's that particular effect that these researchers were utilizing to nucleate and grow dendrites from solution. So in this particular case, they were seeding these gold particles onto the membranes, and in the presence of the electron beam, these dendrites would grow out of solution and form these structures that you can see here. So the interesting thing was what they wanted to characterize in the microscope was actually the growth and propagation of these dendrites over time, because you have the full imaging resolution of the microscope, so you can pretty accurately characterize how fast things are growing nanometer by nanometer. And what they found is that there was a correlation between the seed morphology and the growth rate of these different dendrites. So if a seed was really rough, for example, you might experience faster dendrite growth. If it was smoother, you might experience slower. And they were able to correlate that by doing analysis of the images and videos that you're seeing here after the fact. So, these are just obviously rich data sets showing how things are moving over time, but you're also gaining access to the spatial resolution of the microscope as well. So this is just one example of some nucleation growth phenomenon. In an entirely different field, but in the same sort of realm of interest, is the nucleation growth of mollusk shells. So I didn't know this, but apparently there's over 50 different proteins that are involved that a mollusk uses to take its shell and conduct the biomineralization process by pulling minerals from the outside solution and forming and enlarging its hardened shell. So there's a lot of complicated step and a lot of really sensitive chemistry that goes on. But these researchers isolated one protein in particular that had a pretty profound effect, and they wanted to study that in situ. So this is something that can't occur really effectively without the presence of liquid, because that's important for how this actually occurs in the real world. So they still needed the same things that I kind of mentioned before, replaceable tubing. In this particular case, they were interested in characterizing the effect of this protein specifically on the growth of the mollusk shell. So as I mentioned, the electron beam can locally change the chemistry. So what they did is looked at the solution without the presence of protein, but then they needed to actively mix protein solution into the microscope. And that's what I've shown here is the two chambers that come into the design select holder. That's where you can inject two different liquids if you want, and you can actively mix liquids in the microscope so you can see the effect of a sort of secondary solution that's being mixed in with the primary solution. So what effect that had is shown here. So this AP7 protein was the protein of interest that they were looking at. And by mixing that into a precursor solution, they were able to nucleate and aggregate particles that effectively mimic the biomineralization process in real time. And so that's shown with the video here, with the formation of these clusters that spontaneously generate from the presence of this protein. And then you can see in a little bit more detail in the high angular dark field as these aggregates will actively grow together and form clusters. So this was, there were some other implications for the different types of clusters that they saw, but this is biomineralization happening in the microscope. So you're actively mimicking the real world conditions. And that's
under the effect of electron beam, yeah.
Yeah, of course, I mean, they're imaging for the electron beam, sure. But in general, that's kind of what in situ is all about, is especially environmental in situ is about mimicking the real world conditions, doing operando type experiments. So an entirely different area in terms of characterization is looking at metal reducing bacteria. So this was some research that was done to further investigate a type of bacteria that spontaneously synthesize nanoparticles. This was being explored for green synthesis route of gold and palladium, I believe, nanoparticles. So in order to do that, though, you've got to have living cells that are put into this liquid cell. You need a liquid environment, but you also need to do EDS analysis. And so at Protochips, we put a lot of effort into making our holders compatible and efficient for doing and conducting EDS analysis. Recently, we released a new set of E-chips shown at the top there that have reduced material around the imaging window. And what that allows you to do is get direct line of sight from your sample to the detector, even without tilting it. And so you can generate thousands of counts per second. And this is a 6-time improvement over previous designs. But in general, all that's done so that you don't have to sacrifice EDS capability when you're doing closed cell experiments. So what they found essentially was the decoration of these nanoparticles within and without this, sort of within the cell and outside of the cell in a liquid environment. And so some of this had been known before, but they were able to do EDS analysis showing the gold and palladium, where it was distributed, and sort of how the cell was interacting with its liquid environment. So the important part here is this is the first example of elemental mapping of hydrated biological structures in liquid. And so that's looking at how things are interacting with the liquid environment, especially living things, is really important. And so you can see all of that in the EDS spectra there, and you can see the presence of it in the dark field. So this is just some life science and biological and soft materials. Moving over to something that I think is probably more sort of relevant to the research efforts of the Colorado area from what I found, is studying lithium-ion batteries. So I think all of us are probably familiar with the benefits of lithium-ion batteries: better performance, more capabilities, longer lifetime, that type of thing, but also the tremendous downside of them, which is the limitations of using them for devices due to their propensity of exploding. And so these researchers, this was at the research conducted at P&L, they were interested in characterizing the dead lithium that forms at the nanoscale for this battery, characterizing the electrochemistry of that reaction, and also looking at this solid electrolyte interface layer. So in order to do that, you had to do this in liquid because that's the mimicking the real world conditions. But you also need to do quantitative electrochemistry measurements. And what I'm showing here is what that looks like in liquid. So on our top E chip for electrochemistry experiments, we have electrodes patterned onto the chip itself. And those electrodes are exposed to the liquid so that you can conduct cyclical voltammetry and all sorts of electrochemistry measurements in the liquid environment. So that's all the introduction, but this is actually what they saw. So in the microscope, they were able to identify lithium using Z contrast, because lithium is a very light metal. And there was a lot of research that went into just being able to conclusively say whether lithium was being deposited or not. But the really important part, and I think a really cool part, is seeing the growth of these dendrites and the incomplete dissolution of the lithium as the battery is cycled. So this is the first cycle shown at the top. And what you can see is that not all the lithium goes back into solution. And by the third cycle at the bottom, you have quite a large volume of dead lithium that's being accumulated in the battery solution. And over time, what that can lead to is a short circuiting of the battery. And this is the problem essentially that they were looking at solving. The important part, and kind of one of the cool takeaways of this particular research, is because they were able to do these experiments in situ, they were able to glean new ways of modifying the electrolyte chemistry to actually improve the Coulombic efficiency of the battery. So in a paper that subsequently followed this, they're actually able to increase the Coulombic efficiency over these batteries just by modifying the liquid, all of which was discoveries that were made possible by this particular set of in situ research. There's also a whole webinar about this topic in particular on our website, so if you want to learn more about this, it's free to access and you can go explore that. But yeah, cyclical voltammetry combined with in situ imaging confirmed the irreversibility of this reaction and the mechanisms sort of governing some of that. So, in conclusion, I tried to go through that really quick just in the interest of time. But again, I haven't touched on all of the research areas, and so this is just some other publications and results that are out there. But in summary, there's a lot that you can do by imaging samples in liquid these days. For batteries and other types of applications where you want to do electrochemistry, it's very important that you can quantify that. I've tried to emphasize, and again, mention EDS and the capabilities of that. You already have access to the microscope, making sure that imaging in liquid isn't compromising that. Liquid flow and mixing is really important if you're trying to conduct complex reactions with various chemical compositions of the liquid. But then also I think it's worth noting that you want a system that's easy to use. Grad school and doing this research is hard enough. So we've tried to make everything as easy as possible to use, and we provide technical support and the most extensive liquid cell publication record on the market. So all of which we give to you so that you can be supported when you're trying to do these experiments. So liquid cell experiments aren't the easiest, but we do everything we can to make them as easy, as quantitative, and as insightful as possible. So moving on to our last product that I'll talk about is Atmosphere. So Atmosphere is just like Poseidon in a lot of ways and very different in some. So with Atmosphere, you're trying to introduce a gaseous environment into the microscope. The way we do that is by providing a manifold that takes 3 incoming gases, mixes them together using software control, and then delivers them to the holder in the conditions that you want. And so this is just kind of a flyby of the atmosphere system showing the different parts. So these are your gases. The gases are lined up directly into the atmosphere. The manifold that does the gas mixing. They're delivered up the lines into the holder. And then the holder is controlled and the manifold is controlled by the software. So it's a more comprehensive system, but it's all designed to allow you to introduce gases precisely and safely into the microscope. And just like before, I'll kind of show in more detail what that process looks like. So just like before, you're going to deposit your sample onto electron transparent silicon nitride membranes. Those two chips are going to be combined in the atmosphere holder. And when combined and sealed with a single O-ring, which is shown here, that will safely seal the gaseous layer. So the O-ring goes in place, it aligns your parts, the two chips go together, and they form a seal for the gas that will soon be introduced into the holder. The lid has a cutout here for EDS optimization, which I've talked about a bit. And just like with Poseidon, we embed electrical contacts into the top chip or bottom chip, depending on how you want to do your TEM experiment. And those electrical contacts are there to make contact with the heating membranes so that you can heat samples up to 1,000 degrees in the microscope. So once assembled, the holder goes into the microscope. There's an electrical connection in the back for doing the heating, but there's also gas lines for connecting to the manifold. So those just go directly into the holder. And unlike the other systems, there's a few other things that go on behind the scenes. So this is how the manifold is working. It's taking incoming gases at very high pressure. It's taking them into the system and using automated software controls that are accessible with a click of a button. It's taking those gases, it's mixing them, it's reducing the pressure, it's increasing the flow, whatever you want to do to mimic the realistic conditions that you're trying to imitate in the microscope. That's what the manifold is there for. It takes that gas, it delivers it into the holder, and then you're basically ready to do your experiment. So this is a cutaway of the holder showing where that gas layer is actually going. And you can see those two membranes there where your sample would be sitting, and then the gas is flowing across those two membranes.
What's the maximum pressure there?
1 atmosphere. Yep. So what this can be used for is a lot of different applications, but one really big one is in the field of catalysis. So over $20 billion is spent in the field of catalysis each year. And in particular, I'll talk a lot more about automotive catalysts, but in general, a lot of these catalysts are very expensive. They're using precious metals. They're nanoparticles, so they're very, very small. And there's very complex reaction pathways that they go to take an incoming solution or gas and changing it to a different byproduct. And in general, I like using the metaphor of the sort of black box. So in catalysis, you've got a lot of different things that are happening. You can only oftentimes see what's happening, things coming in and things going out. So there's a lot that can happen in that black box. And when you have very delicate chemistry, you have very sensitive samples, you have very expensive alloying elements, and you have things that are very, very small, there's a lot that can go on that you don't have access to. So in situ allows you to sort of peer into that black box and see directly, atom by atom in many cases, what's happening with these catalysts in real time. So in this particular research, they were interested in synthesizing nanoparticles with different characteristics. So in order to do that, atomic resolution, because they're very small, pressures up to an atmosphere, so they can mimic the real world, and then they needed a stable heating membrane that they could maintain at high temperature without degrading over time. And so what I'll go through here is just kind of what they found. So they looked at 2 different annealing phases that they could conduct in the furnace, but they were able to do that directly in the microscope and see the interaction of the platinum, which is the really catalytically active element, with the cobalt oxide interior of these nanoparticles. And so there was a few different temperatures that they looked at, a high temperature and a low temperature. But the important part here is that the surface of the nanoparticle needed to be enriched with platinum in order for it to work more effectively. So they did this initial high temperature annealing phase, but what they found is when reducing the temperature to a lower temperature, they were able to see kind of the growth of this platinum shell occurring atom by atom. So this is at 300 degrees C, and what you can see is these monolayers forming bit by bit. And so this was all designed, all used to investigate the synthesis routes by which these catalysts could be created. So that's synthesizing catalysts, and in a totally different research, they were interested in watching how those catalysts evolved over time in the presence of a different environment. So this is sort of more true catalysis in the sense of how things were reacting with their environment. And this particular research effort was looking at a phenomenon that had been known for a long time, but had never actually been directly observed, and there was still a lot of unknowns and uncertainty about it. And it's a phenomenon called the strong metal support interaction, by which a support can actively interfere with the chemistry going on with the catalyst, and can make things better, it can make things worse, but it was known about, but it was not necessarily understood or quantified or observed directly. In order to study this SMSI reaction, though, they needed to deliver realistic gases that mimic the real world, and they needed to be able to control the environment precisely. So recently, we've overhauled our atmosphere system to allow it to accept a wide diversity of gases, and it'll allow it to mix those gases in pretty much any composition you want. So whether you've got a complex multi-component gas that you're using for your research, or whether you're using pure gases, mixed gases, argon, helium, carbon dioxide, atmosphere can handle all of that, and it can actively mix those gases together if you want to control sort of relative ratios of the different gases. And I just showed a screenshot here of the Clarity software, which basically has a button for every single variable that you want to control. So whether that's temperature, whether that's flow, pressure, mixture, all of that is accessible with the button in the Clarity software. I should also mention everything I'm going to show here, which is only going to be one slide, is also a webinar that is free to access on our website. So you can learn a lot more about this particular reaction. But essentially, what these researchers were able to do is use different gas compositions and observe this SMSI, this interaction occurring in real time in the microscope. And so what you can see is the same nanoparticle under three different environmental conditions, exhibiting three different structures. So on the right is a pristine nanoparticle that has no, it's just palladium exposed to the outside environment. And under certain conditions, you can form an oxide monolayer that's amorphous, that interacts with the environment in a different way than the pristine nanoparticle. And also, in a different environment, an oxide bilayer that's crystalline that has an even different interaction in terms of the catalyst reacting in this environment. So this is visibly the SMSI observed in real time in sort of atomic resolution and pristine detail. So that's watching how a catalyst is evolving, but taking that one step further is actually characterizing the performance of a catalyst. So like I said before, ultimately a lot of these catalysts are, the efficiency of them is governed, or is their efficiency and effectiveness is their ability to take an incoming set of conditions and turn it into a different one. The efficiency of that governs the performance of the catalyst. So in this particular case, these researchers were interested in the methanation process by which CO2 could be converted to methane and water vapor. But in order to do that, to characterize the efficiency of that, you have to be able to characterize the outcoming gas and see how it was affected by your catalyst in situ. So recently we've worked on atmosphere to make sure that it's compatible and works well with RGA and mass spec type equipment. And you can see schematically what that looks like down here, which is a flow control system and a connection directly of the outcoming gas line to a mass spec. And so what they were able to do, I'll show in a minute, is use that mass spec to correlate what the incoming gas was with what the outgoing gas was. What I'll show you here is just a sort of different step that they conducted to look at these catalysts, but they were also interested in characterizing the presence of nickel nanoparticles that would spontaneously nucleate and actively engage in the catalysis reaction, all of which occurred only at high temperature. So long story short, this is sort of the result here. So they introduced an 80% hydrogen, 20% carbon dioxide gas, and that was a constant gas that was flowed into the cell. And what you can see is over time, and as the temperature of the catalyst was increased, you can detect more and more presence of methane and H2O over time. And so as you're increasing the temperature of the catalyst, it's becoming more catalytically active, and you're actively able to detect the generation of new gases from the catalyst and the transformation of these different chemical elements in situ and in real time. So not only were they able to characterize the performance efficiency of these catalysts and see the effect of temperature, but they were also able to image the morphology, the structure, and the chemical makeup of the nanoparticle catalysts in real time. And so the nickel nanoparticles, which are highlighted out there with arrows, were really important in governing some of the reaction kinetics that were occurring in the microscope. So using mass spec can be really powerful for characterizing not only the, atmosphere can be used to characterize not only the structure of the catalyst and see how it's evolving, but you can attach mass spec and you can characterize the performance of your system as well. So it's really a multi-dimensional data set that has profound impacts and implications for studying catalysts in the microscope. So the last application I'll talk about is corrosion. So in this research effort, some researchers at the University of Manchester we're interested in looking at steel pipes and how they corroded in high pressurized water reactors in nuclear power plants. So those are really caustic, really aggressive conditions. They wanted to mimic that and see how these samples were corroding over time. In order to do that and characterize the stress corrosion cracking, which leads to pipe failures shown at the top right, they needed EDS analysis to characterize how the alloying and desegregation was occurring. But they also needed to be able to study these samples in the presence of water and water vapor. And we worked really hard, again, on atmosphere to make sure that all the pipes, all the tubes were compatible and capable of handling water vapor and other gases. And so what they did is they heated this sample up over time in the presence of nitrogen gas and water vapor. And what you can see is the elemental analysis over time showing the segregation of various elements within the steel sample. And what you can see, what you may be able to note here is that chromium in particular, which has a really profound effect on corrosion inhibition in steels, starts segregating away from this grain boundary that they were imaging over time. One interesting thing about this particular experiment is what you'll notice is the sample is not necessarily tremendously high resolution. And the reason for that is they kept the sample deliberately thick so that they could do a post-mortem analysis to further interrogate the effects of this desegregation occurring over time. So after the experiment was finished, they did a good cross-section on the sample after it was taken out of the microscope. And what you can see is further segregation of chromium along the grain boundary that occurs at different depths within the sample. So again, chromium is has really strong implications for how a steel can reduce and resist corrosion. And the chromium reduction was from 16% down to 4% along the grain boundary, effectively, showing how reduced the corrosion resistance was. So this was also the first study to directly observe all stages of the segregation occurring over time, and that's done with elemental analysis in situ. So just in conclusion here, I showed a few different types of research efforts that could be accessed with atmosphere, but there's a few things that we always like pointing out that are important to consider. A heating membrane that's going to be able to heat your sample up without changing the chemistry of what it is that you're studying. A design that allows you to do EDS analysis so you can track the chemical composition over time. The ability to control your environment is really important. So looking at high pressures and low flow rates and vice versa. And then the ability to control the environment to mimic real world conditions, so diversity of gases, looking at water vapors, things like that. Of course, this is the same loading design as the Poseidon system, which I think I've already mentioned. We put a lot of effort in making that easy to use. But yeah, I think just to wrap up here, I've talked a little bit about our products, but I hope I've given you just a glimpse into perhaps why so many people are turning to in situ these days to characterize and experiment on their samples. So their in situ results are no longer these finicky, difficult, irreproducible experiments. They're truly quantitative and insightful ways of exploring the chemistry of your sample. And I hope I've also shown some subset of all the different types of dynamics that you can visualize and see directly in the microscope. The last thing that I'll mention is everything that I've shown here is not research that I've conducted or Protochips has conducted, but is conducted by researchers and customers from ours around the world. So none of this would be possible without their support, and we see ourselves as just one small piece of the research efforts that are quite impressive. So we like giving talks on behalf of our customers. We like them to give talks on behalf of us. We like featuring their work. We like having user meetings where we get to interact and engage with them and connect them, because the growth of this in-situ market has been really, really cool to be a part of. And we couldn't do that without their support. So I always like thanking them. And of course, thank you guys for having us. If you have any questions, Steve and I are here to answer them.
Thank you very much. Thank you. Thank you very much.